Hello, my name is Cameron Young. I am a junior majoring in clinical neuroscience, and this is my neuroscience video presentation. So the research article that I decided to use for this project is the gigantocellular reticular nucleus and how it plays a significant role in locomotor recovery after an incomplete spinal cord injury. This slide was just included to show some additional information about the article, including where I received it, the section that it was in, the date it was published, and its authors and editors. So just some background information on partial spinal cord injuries. For decades, it has been traditionally unacceptable, or people didn't think it was possible for someone to recover from a partial spinal cord injury, stating that the adaptation wasn't possible for motor function. But in recent years, it has been shown that spontaneous recovery of crude motor function has been seen in both humans and animals in their legs or their hind limbs. And this includes injuries where a very limited number of fibers remained. It has been shown that there are two modes of neuronal outgrowth that's noted after a partial spinal cord injury that is a possibility in assisting the return of motor function. And those are the local rewiring of lesioned axons, so the ones that break from this injury rewire themselves, or fiber growths that were spared in the injury branch over and fix the injured area. So it has been shown in rodents such as rats that the bulbospinal projections can generate both of these modes, and that these modes can come from the same nucleus in the brainstem. And then our other two modes, which is that midline crossing NRG fibers, the ones who are compensating, and then the locally rewiring injured axons, which are the ones that fix themselves. The main purpose of this experiment is to investigate those locally rewiring ones, so those injured ones that are fixing, versus those axons that will reach over and compensate, to see which ones help specifically and which ones don't help as much. And we do that by using special projection-specific genetics to introduce a silencing HM4DI receptor to the axons, neurons, to get specific genes and get specific NRGs. So the hypothesis that the authors had for this research article was, if the rats are given partial spinal cord injuries, then it will be shown that both compensatory and locally wired reticular spinal plasticity will contribute to recovery of walking, wading, and swimming. So it's best if they are both used together as far as recovery. So this is just a quote from the journal. Moving from a correlative to a causative understanding of the role of neuroanatomical plasticity for functional recovery is a fundamental for the success translation of treatment approaches from experimental studies to the clinics. So that's basically saying with these experiments, with these testings, we can hopefully move from rats to humans and help humans who have partial spinal cord injuries. Now that we have all the background information we need, we are going to go straight into the experiment itself. So there is a very complex method here, so I'm just going to simplify it as much as possible. We're going to have 20 trained rats who are trained to walk, wade, and swim on this locomotor that will help analyze their methods before and after partial spinal cord injury. These rats obviously properly taken care of. These rats will be put under anesthesia at one point and be given tattoos on their shoulder blades, their shoulder joints, their iliac crest, and their hips. Then they will be penned in at their wrists, at their fifth metacarpal head of their hand, the tip of their fourth finger, their ankle, their joint of their fifth toe, the tip of their fourth toe, and then the tail at the base, 1/3, 2/3 up, and then the tip of the tail. They will be monitored daily before they are put under anesthesia again to get an adeno-associated virus injected into them, to in a craniectomy to recept the DREADD receptor through HM4DI, and that's going to be injected 3 times with 5-minute breaks in between. It's going to be 50 nanoliters each. Then they are going to be put under again, and they're going to get a laminectomy between the C3 and C4 vertebrae, or the T12 and the T13 vertebrae. It just depends on the rat. Those are going to be 8 injections with 100 nanoliters each, and they will be spaced by distances of 500 micrometers. So after that, they're going to have locomotors 3 weeks after the injections, and for walking weighting, they're going to be on a treadmill. With the weighting, the water is going to be 5 centimeters high, and then with swimming, there's going to be no treadmill, but the water is going to be 24 centimeters high. Videos of these rats must be in a straight line with a constant speed, and they need to have a cycle of 3 to 4 steps for walking or wading, and 5 to 6 cycles for swimming. And their legs and feet have to be visible throughout it to make those calculations. For those rats who have the DREADD injected into them, 30 minutes before these tests, they get 10 milligrams per kilogram of CNO, and then that is in a 45-minute window, so you know that the chemical is the cause of that. The best video is taken from each animal after all the trials. The walking and wading will have an onset of the first forelower contact to an offset of the MTP liftoff, and then the swimming will have a onset of the minimum angle between the knee and ankle, and then the offset is maximum extension. And this is just a little photo of where the injection sites would be for the experiment that they did. So this is just a photo of what the glass tank where they are recorded looks like. This is an example of when the mouse is swimming. So these white marks here are the mirrors, and they are used for parallel recording, so they can see all angles of the mouse. And the limbs are tracked by the tattoos and allows that calculation for the kinematic parameters. So this is the results for the walking based on the control trial, the servo trial, and the lumbar trial. Each trial shows a difference in stability, speed, and timing aspects, and this is showing that the silencing of these NRG neurons resulted in a big reimpairment of that stability, speed, and timing aspects when in the recovered walking position. Similarly to the walking, the wading in the water also had a similar effect, showing that the recovered times with both of the modes that we mentioned previously had the best outcome. And it also stated that this one used more of that compensation axon than the fixing of the injured one. And that was just shown because wading is showing the repairment of that speed and agility, and it's just a different maneuver, so those neurons worked better in that case. Finally, for swimming, which resulted in only one feature because of the limited features that are used, it was classified as Class A, which means best possible outcome. And the CNO-induced silencing showed reimpairment of recovered swimming function, especially under conditions where the compensationary NRG was silenced. So this is basically saying that with all 3 of these functions, yes, one mode is better than the other, but with all 3 of these functions, you're going to need both to properly wade, swim, and walk. So just to end our experiment with a discussion, it was showing that 19 out of the 45 features in walking, 9 of the 45 features in wading, and 1 of the 18 features in swimming had nearly full recovery with both. Additionally, there was localization needed for full swimming execution, so you needed both of those modes to be able to do all 3 properly. In conclusion, this shows that both modes of the axons are needed to have as much motor sensation returned as possible after a partial spinal injury. And these were my sources that I used. Thank you very much.